That's beautiful. Okay, we're recording. Sorry about that, everyone. Oh my gosh. I feel like my grandma could have done that a lot better than I could just then. Welcome. So excited to have you guys all here. We're going to jump straight into it. Welcome to Conservation Diver, Coral Taxonomy and Identification. You guys may have seen on our Facebook post that when we were, sharing this online, that we sort of stated that me or Elle has some weird ways of remembering things. Now, if you guys have read through the PDFs I sent you, you may think, what the hell is one of the sheets with names of corals and some very weird sort of sentences next to them. For me personally, I find mnemonic devices very useful when it comes to remembering taxonomy. I'm going to let two people in. This is wonderful. I find mnemonic devices very, very useful when it comes to remembering coral taxonomic names, because for me personally, I just think that Latin is hard. So I don't know about you guys, but I don't know that many people that speak Latin today. And when we are talking about taxonomic terms, most of them do have Latin origin. So by creating sort of little word associations and linking them in my head that way, I find it a lot sort of easier to get around these sorts of topics, okay? So as we go through the slide, I might sort of remind you guys of some of those. It doesn't work for absolutely everyone. When I first started teaching taxonomy, I didn't necessarily use them all the time. And then one class I did, and people thought, hey, well, people reflected and they told me, they said, hey, some of those were really weird. But strangely enough, now I remember that coral. I know that that's the one that looks like an earlobe or, something like that. And it is this sort of interesting way to remember things. For me, the other thing that I personally do to remember new Latin taxonomic names is often I'll just write them down over and over again, which I know seems a little bit boring. But if you are one of the people that really do need to or want to get around this sort of topic, and it's important to you in your career, it could be anything biology really, or anything that involves, a new language that isn't, I guess, sort of layman's, then I would recommend trying it. So for me, when I am learning new names, I write them down over and over again. Here's some of the pages of some taxonomic names I was just trying to learn myself for some gastropod species that I was totally unfamiliar with. And you kind of do seem like a little bit crazy writing them down over and over again, but for me, it really, really works. And I've gotten some of my students to try it and they sort of say, it's annoying and it's time consuming, but you do get them stuck on your head once you've had to go through that motion of just copying them over and over again. The other PDF I showed you guys was just some extra resources that I would very highly recommend. WORMS is the World Register of Marine Species. And as some of you guys may be diving into taxonomy more and more as your careers or studies progress, you'll notice that a lot of names within taxonomy, especially within the marine world, are sort of getting juggled around. And I don't mean juggled around, pretty much we've recorded things as one species perhaps in the past and used morphological features to do so. And as technology is advanced and we might be gaining more access to genetic testing, we're realizing that we've made maybe a few mistakes in the past and we can more accurately place them in the phylogenetic tree, which is really awesome. So World Register of Marine Species is amazing if you ever want to double check a name, check that you've got the spelling, see its relatedness, see its classifications all the way up to phylum. So very useful. Another one that I highly recommend is the Coral Finder. I've got one myself and it's my favorite book ever, okay? This is just, it's the shit. It's so good. And it's incredibly easy to use. It's color-coded. So if you are in the Indo-Pacific, I would recommend getting your hands on one of these. They're a bit expensive, which is annoying, but I actually would say it's worthwhile. But a lot of centres that do sort of a lot of, conservation work and education will often have a copy or a few copies of this book. And so they should. So yeah, also after this lecture, if you guys have any questions related to coral taxonomy, if you took a photo on your GoPro and you want to help ID it, send it through. I love this stuff. My email is at the bottom of that PDF sheet. But also my email is just l@conservationdiver.com. So it's pretty easy to remember. Anyway, so that was our introduction, I suppose. Now let's get down to it. Can everyone still hear me? Give me a thumbs up or like one of those, reactions. Awesome, cool. getting them from a couple of you guys. That's fantastic. So welcome, coral taxonomy and identification. So first, I just want to express why this is actually an important and essential part of conservation. It's more obviously an essential part of monitoring. When you monitor things, you know, being able to record them to the most amount of detail that you can is obviously more ideal than having less detail. But how do we apply that to conservation? That's obviously something that we, a conservation diver, are doing. We're applying active restoration to our work. So I've just got a question. I haven't changed slides yet. I'm so sorry. Who was that? From, oh, it just says Zoom user. I am just still on the first slide. So I took a while to introduce everything. So your screen's not frozen, I assure you. But yeah, so essentially, when it comes to applying taxonomy of corals into conservation, it's really important to know what corals you're dealing with, as opposed to saying, hey, we've got this reef and there's high coral cover. It's good. We don't really need to do anything. What having an understanding of coral taxonomy can help you with is understanding the level of diversity which exists within, you know, your restoration area, whether that be one bay or one island and so forth. Well, I used to work, which was on an island called Koh Tao in Thailand, which I know that some of you guys have been before. Our base was in an area called Sherlock Bay, and it had really, really high coral coverage. And a lot of people thought, you know, what's the need for restoration here? There's all this coral, you know, what's the problem? But the fact is it was pretty much monospecific stands of Acropora and a few other specks of different genera floating around the place, but it didn't have a lot of diversity. So it's actually very susceptible to coral diseases and also storm action and other events like that. And often we did find outbreaks of diseases and coral predators within this bay. But yeah, so that's just one very simple example. That being able to understand that, you know, coral coverage is one thing, you know, 100% coral coverage is good, but if it's 100% coral coverage of a single species, then you might not have a very resilient reef. So also today, oh, I'm just going to admit someone. Oh, Brad's back. He must have dropped out there for a moment. Awesome. So, oh yeah, that's right. So during this lecture, we are going to mostly focus on identifying corals to a genus level. And that's because the genus level is quite reasonable level.
Hey, how you doing? You all right?
I'm good, I'm good. How are you?
Yeah, good, thanks.
Oh, someone's not on mute. Okay, so, oh yeah, so when we're making in the field identifications, often it's quite realistic to be able to ID them to a genus level, whereas to a species level can be a little bit tricky. You sometimes might need genetic samples, or you might need a skeletal sample and a microscope. So we do focus on IDing things to a genus level because it's just more realistic. And of course, getting to the most specific level of diversity or, you know, of taxonomic classification is better than just saying hard coral or, you know, the branching coral or the bouldering coral. So yeah, any questions so far? If not, we'll actually properly start getting into the lecture. There we go. Cool. Yeah, there we go. So what are we focusing on today? We are focusing on hard corals. So if we look at this sort of graph here, we can see at the top of the phylum which hard corals fall under is the Cnidaria, the Cnidarian phylum. And this includes other animals such as your hydroids to your, oh, that's right, I've got my pointer. I don't have my pointer. Laser pointer. We've got our hydroids, as you can see to the far left. So these are very small animals, generally speaking. Often they'll grow on things like mooring lines. Sometimes if you ever, you know, maybe held onto a mooring line on your safety stop and then got to the surface and your fingers were a bit stingy, then that's because you were probably actually touching some hydroids by accident. You can call the Cnidarian phylum the stingy phylum if you really want to, because, you know, nematocysts or stinging cells are one of the things that all of these animals that we're talking about have in common, okay? We've also got different varieties of jellyfish. These also fall within the Cnidarian phylum, okay? If we go to the next step, we are looking at the Anthozoa, okay, which have our hexacoralia, which includes our hard corals or sclerotinia. Anthozoans also include Aceriantharia, which has tube anemones. Sometimes you find them in silty muck ecosystems. They kind of do look like tube worms, often they're called. And then also your Octocoralia, which is your soft corals, which are identifiable as having 8 tentacles per polyp. Okay, this also can include your gorgonians and sea pens and blue corals as well. Blue corals not being blue, but having blue skeletons. All right, so next step, we do have the sclerotinia, and within them, within the Hexacoralia subclass with the sclerotinia, we have Zoantheria, which is like your zoanthids, which we're going to look at in a moment because they can be easier to mix up with hard corals. We've got a corallimorphs, also a little bit similar looking. So we'll look at some photos of them in a moment. Our Antipatheria, our black whip corals, this is underlined because it's currently being revised. So Antipatheria hasn't sort of been confirmed by, you know, the best in the field of taxonomists for that particular group. But we've popped it in there because thus far it has been quite official. And the Actinaria, which is our sea anemones, okay? So let's go a bit further. So within the order Sclerotinia, we've got 35 families of corals. Within that, there are hundreds of genera, and within them, there are over 1,300 species. So again, just for the sake of practicality, IDing things to a genus level as opposed to a species level is also a bit more realistic in terms of just, you know, the size of your brain and how many Latin taxonomic names you might actually be able to fit in there, okay? So let's just quickly recover or go over coral skeletal anatomy, which I'm sure a lot of you guys will have a basic understanding of, at least if you've done maybe any of our courses or if you just have a general interest in corals or coral reefs at all. So we have the corallites, which you can see I'm pointing to the real photo here, but also this and this and this are all different variations of a corallite. You can think of it as the cup-like structure in the coral skeleton that the polyp, that being the living animal, sits on top of. And they're, you know, layering down calcium carbonate and pretty much thickening this up as they go. But the corallite is where all the action is, okay? It's where all the important stuff is. We've got the columnella, which you can kind of see, I nearly pointed it with my fingers then, instead of my, my pointer, which is the central part, essentially where the mouth is going to be sitting of each of our corallites, okay? Then we've got the sinustrum. So not all corals will have one. What this is a fancy word for is for space in between the corallites. So the sinustrum sits in between the corallites if there are indeed separate walls. And while walls aren't technically labeled here, you can sort of tell and guess where the wall is going to be. On this, you know, this is the edge, this is the wall, and each of these are also walls here. You can think of this, the septa are sort of like the ridge-like structures that run from the columnella all the way up to the wall of each corallite. And then the costae are the ridges that run down on the other side. Occasionally, we're going to have corals where the walls are joined, which means we're only going to have septa and there is no other side. You'd just be dropping down into another polyp. Don't worry if that sounds a little bit confusing because we're going to be seeing plenty of examples of each of these throughout this lecture. So don't worry too much. At the top, if we have a nice flat rim, it's called the theca. That's kind of, you know, quite a distinct, nice wall. Sometimes we don't have that sort of distinct theca though, and it might just simply be that the septa run continuously until they turn into the costae, in which case they've called it the septocostae, which is probably like the one time in science where they kept it simple and they just called it what it should obviously be called, which is the joining of the septocostae. Yeah, so yay science. That one was an easy naming, I suppose. So then we've also, what have we missed here? Oh yeah, sorry, I forgot to point to the real life photo here. So this is a septa on the inner part. On the outside, you can see the jagged nature here, the outside structure of those walls. Pretty straightforward, relatively speaking. You guys might see down here, there's sort of like in this photo down the bottom, or this diagram I should say, sort of looks like a secondary wall or a little sort of like crown that sits just over the edge of the mouth or over the top of the mouth. And that's called the paliform lobe. Not all corals have this, But when they do, it's usually pretty important because they look like other corals. And you guys will know what I'm talking about when we get down to some of our Merulinidae. Sometimes the difference between two corals might be the fact that one has a paleoform lobe and one doesn't. But again, all of these terms, don't worry, you don't have to remember all of them, right at this moment, because I am going to be pointing back to them and referring to them when we're actually looking at our corals. So pretty easy. We have two types of budding in corals. When corals aren't getting their sexy time on during the special spawning times each year, they will be breeding asexually. So every coral colony does start as a single polyp that is produced sexually during spawning seasons when eggs and sperm are released into their water column. They mix together on the surface because they are buoyant, and eventually they settle down, turn into a single polyp. And that's the end of sexual reproduction for probably the next 7 to 10 years or so at least. From that point forward, they will asexually reproduce, where a single polyp will turn into more. So intratentacular budding is when you pretty much have a single polyp, like you can see on my hands here, and they bud off like this, sort of turning into a figure 8, and they pop off from one another, okay? Think of intratentacular as internal, it's happening from what was already sort of existing there, if that makes sense. And then we've also got extratentacular. Think of that as external budding off of the polyps that were already there. So they're just doing their own thing, these baby polyps. You can see them here, these smaller polyps, but just simply sort of pushing themselves up in between these larger ones, all right? And here you can really see that figure 8 shape going on. Again, remembering that while these are two very different genera, this is a Diploastria Heliopora and this is a Dipsastria, you can sort of still see those structures talking about before. Here's the whole corallite, the wall, and again, here are the corallites, okay? But again, intratentacular and extratentacular budding. Sometimes this is very useful to remember because certain corals, of course, have intratentacular budding and some will have extratentacular. Any questions so far? Remember that you can do those hand things or like whatever if you guys need to. So just do it if you need to, but otherwise I'm going to keep going. We are going to get, of course, into the taxonomy, but this is just some of those essential like key knowledge points that it's good for you guys to know before we get into it. We'll fly past this bit because we're going to sort of be repeating it during. You can see the real life photos obviously up to the side here. I'll sort of focus on this side here. In terms of colony formation, corals do grow in many different forms. So we do have solitary attached, and we have a single polyp, a single individual corallite, and they remain attached and they don't actually bud off. Some corals are not colonial, and we'll get a few examples of that throughout this lecture. We have the solitary fungoid, which is again a singular polyp, but they are not attached. So often people refer to these as your mushroom corals, which you guys can see along the side here. And they're some of my favorites, they're awesome. Then we've got the plocoid, we've got very distinct walls and separate polyps. We've got the nice, in this situation, we've got the nice anastrum. We've got ceroid, where they are budding head to head here as well. And then we've got basiloid, where they are quite tubular in nature. You could describe it as too. And our equivalents over here. So then we've also got thamnasteroid, which is an interesting one. It's ones that have no distinct walls. So this is pretty cool. We're going to be having one of our corals that we're covering today, or a couple of them, such as Pavona, which is a very, very common coral of the Indo-Pacific, which takes this growth form. There's actually a photo of it just there. So it is indistinct septa flowing between polyps. They're kind of flower-like. There are no costae because there's no wall that's being met and the costae coming down from the other side there. All right. So then we've got meandroid, which is a pretty easy one to remember. Meandroid is meandering, that type of coral. We've got meandroid pachyceres type, which is sort of a variation of meandroid. There is a genus called pachyceres, funnily enough. That's just doesn't quite have a colony formation. So this is sort of to describe that genus. We've also got flabellate, which is practically, it's meandering in nature. However, we have these sort of gaps in between. And if you ever saw a coral like this, sometimes it looks like they're a little bit bit broken, but it's just that they've got these sort of sometimes jigsaw-like pieces that kind of half sort of fit together, but you will have sort of gaps in the middle, a flavonoid as well. And then we've got Hydnophoroid, which similarly to Meandroid Pachycereus type, describes a single genus of coral called Hydnophora, which has these bumps along the skeleton called hydnophores. So there's not a lot of variation in what they call this coral, its bumps and its colony formation in this example there. But this is an actual photo of the Hydnophora. It is a very, very beautiful colony, that's for sure. So what looks like hard coral but isn't hard coral? So when I'm teaching my students, you know, and if things are new to them and they might not know what everything is, I always say, you know, try not to be invasive, of course, but if you're really unsure about what something is, giving it a little bit of a wave can actually help so much in helping you come to the conclusion of what that thing is. So when it comes to corallimorphs, they've got these little tentacles here. Corals, again, are cnidarians. They're related to jellyfish. They've got stinging cells called metacysts, and they've got little tentacle-like structures. These guys sort of do too. They're a little bit like more short here, but you can see them speckled all around the edges, and they've got a central sort of mouth area. So someone who might be a bit newer to this might very well go, is this a hard coral? But if you were to give a little bit of a wave, they only have a centralized attachment point, and the edges of them are quite floppy, is probably the easiest way to describe that there. And then very, very easily you can probably say, okay, well, it's not got a hard skeleton. Something about hard corals is that they have a skeleton. They're hard. So they can at least help you work out what it's not. You know, in this case, you can say, even if I don't know at this stage what a corallimorph is, I can tell that it's not a hard coral. Okay. And then we've also got zoanthids. These guys in particular look like a few different genera of hard coral. They're encrusting, they're often colonial, and they look like some of the genera such as Diploastria and Montipora, some of the corals we're going to cover today. But again, the name of the game, if you're unsure, give things a little bit of a wave and see how they respond. And in the case of zoanthids, firstly, morphologically, they're different because they have a double layer of tentacles compared to one, which is what hard corals have. If you want to get up real close and check it out, you can see that. But secondly, what happens to like these entire like polypy shapes here is that they think, you know, is that food coming? You know, what is that water action? So they'll close up, whether it be that they're trying to catch food or they're trying to protect themselves. But you can pretty much see the softness of them when they do that. And they do often close up entirely into like, you know, all these neat little pockets. So even if, again, you don't know what a zoanthid is, you can go, you know, I don't think that's a hard coral at least. Also, guys, I would just like to mention, because I didn't mention this at the start, why are we focusing on the taxonomy of hard corals versus soft corals or versus corallimorphs or zoanthids? None of these other organisms are not important, but one of the things that they have or that they don't have that hard corals have is a calcium carbonate skeleton. And when we're talking about, you know, tropical reef ecosystems in particular, what these ecosystems are really reliant on is having hard substrate, because marine animals, you know, they don't shoot roots and secure themselves into the sand like land plants and things like this. They are encrusting animals generally, at least when we are talking about our sessile animals here, you know, like your soft corals, hard corals, anemones and things like this. They need to have something hard to sit on. So if we have a sandy area and, you know, coral spawning has just occurred and all the gametes are looking for a place, or all the fertilized gametes are looking for a place to settle, they won't probably be able to settle on the sand because it's not secure. They're not going to be able to set up a centralized polyp and then asexually reproduce there because it's just too mobile. It needs something really solid. And while hard corals produce a hard thing, being that it is their skeleton. So hard corals play all these roles in terms of providing habitat and food source and so on when they're alive, but when they're dead, they're also really important because even though the living tissue on the outside, all the living polyps will die, the hard stuff, being their skeleton, remains behind. So maybe that means another coral will settle there and grow into a brand new colony, or a zoanthid will settle there, or a soft coral will settle there. And all these things play these really essential ecosystem roles. And that's why we do focus on getting extra information in a hard coral. So us at Conservation Diver, when we do our ecological monitoring program surveys, we collect at least genus level on every coral along our transect lines, so that we can see diversity and if there are diversity shifts throughout time. So I hope that provides a little bit more clarification for why we're doing this, because I should have done that on the first slide, but it's been a while for me. Next, Nanipora. So I just said that only hard corals produce skeletons. Well, I didn't say only, but I said hard corals produce skeletons. There are some very specific examples of soft octocoralia, which do it too. So Nanipora is a really cool one, and we at Conservation Diver love it because we were the first ones to discover it anywhere in the world apart from Japan. Yay! One of our co-workers, Pao, found it on Koh Tao and thought, this is weird. This is a soft coral, but it's got skeleton. This is unusual for this region. And indeed, it was Nanipora, which previously had only been described in Japan. So we expanded its geographic range understanding pretty much. And while it produces a skeleton, it actually is very insignificant. It's a very thin skeleton. But we can tell that it's an octocoral here because it's got 1, 2, 3, 4, 5, 6, 7, 8 tentacles. Hard corals, guys, Sclerectinia, whether we're looking at a big polyp with tentacles or something very small that you've got to get up close to check out, they always have tentacles in multiples of 6. So whether it's 6 tentacles, 12 tentacles, or 24, it's always going to be in multiples of 6. Like, unless a predator has come along and taken a bite out of one of them or something like that. But the standard is that it'll be multiples of 6. All right. So then we've got our black whip corals. I think these are not too, they're not too easily mixed up with as hard corals. So we can kind of skip past that one. But if you guys hadn't seen photos of these whip corals, this is what they look like. And they generally live in slightly deeper areas, maybe ranging, your 10, 15 meters plus. Anyway, now let's look at the one soft coral that actually is very, very cool. Sorry, they're all cool. But the one that's really cool and being the exception, in terms of producing a calcium carbonate skeleton. So Heliopora is a genus of soft coral that make huge columnar sort of structures, okay? It's been really an interesting genus. Heliopora was monospecific until a very cool lady by the name of Zoe Richards, who's based in Australia, described a couple more species to the genus, which is very exciting for coral lovers in general. But for any of you guys that may have been to our site in Indonesia, maybe you visited Leon, which opened up in 2019, Leon saw Zoe Richards' paper and thought, I always thought there was something wrong with all the Heliopora we have around Gili Air here in Indonesia. And he took some photos and sent them to Zoe. And then he took some samples and sent them to her. And he found out that the dominant reef builder around the island he was living on was a species that hadn't been described to science at all up until like the year before. I think it was 2018 that these new species of Heliopora were described. So he and Zoe have actually published that now. He also found it in the Maldives on previous work that he'd been doing there. But it's pretty exciting. And obviously it is a very big exception to the rule. Hard corals are hard, soft corals are soft, except for sometimes, because science and nature don't like to play by the rules. They make their own rules. And while it's really annoying to us, it's better to just try and appreciate it rather than get frustrated by it, I suppose. But it's really, really cool. If you're ever unsure about if something is a hard coral or a Heliopora, look up close. I've never seen any other coral with such a sort of fuzzy nature to it, okay? So you can see, especially in this box here, the tentacles are out and they're so sort of thin and fine and fuzzy that I probably wouldn't be able to tell in the field if it's 8 tentacles or a multiple of 6, but it does have this very, very fuzzy nature to it, and small polyps too. Excellent. So hard corals. Sorry, I've already been yapping on to you guys for a while, but now we're finally getting to some of the more common Indo-Pacific families. We're going to be covering the families that are white, okay? There are more, of course. If you guys want more information of any of these others, you can feel free to message. But pretty much in this lecture, we could go on forever. Well, not forever, but we could go on for over 300 species worth of corals, because that's how many in the Indo-Pacific. Within the Caribbean, sort of Atlantic region is closer to 70. So most of the diversity exists within the Indo-Pacific. But I can't keep you guys here all day / all night. So we're going to focus on what our team has pretty much noticed over the years are sort of the more dominant, really kind of useful ones to know, especially if you are wanting to monitor and implement conservation practices in your local area, or if you plan to be doing this kind of work in future. So let's start with Acroporidae. So all of our families are going to be blue, okay? If I have plastered a star next to it, what that means is that I think this should be high priority on your list to learn, okay? This, you know, the genera that we're going to be covering today that have a star next to the name, I've noticed are frequenting the literature so much. So if you really do want to get into the sciences of coral taxonomy and so on, I'd say this is, you know, these ones are particularly useful to have a good understanding on. What it means if I've plastered a C next to it is that this genus also occurs in the Caribbean. Just for you guys, because obviously we do have people from all around the world. Sure, the Indo-Pacific has more diversity. It doesn't mean that we're all going to live and work in the Indo-Pacific, okay? So that's just what those codes mean, just so we're all aware. Acroporidae is the family name. Acropora is our genus name, and it's got a lot of species, okay? We're going to cover the species names in the Caribbean in a moment because there's only 3. In the Indo-Pacific, knowing that there's a lot is good for now, at least, okay? So how do we identify Acropora? Main identifying characteristic is that it has what we call axial polyps at the tip of each branch, and they are surrounded by smaller radial polyps. Radial meaning surrounding. So quite literally, radial polyps are the ones that kind of surround the tips of each branches, you know, such as here. So just so you guys can see, this is an axial polyp here. So is each of these, as we can go down the branch. This is a corymbose acroporid, okay? So that means that it's quite a dense branching coral. This is what we'd call just a standard branching coral. And you can still see that quite obviously you've got the tip of each one, which has got this rather distinct and large axial polyp surrounded by all these smaller bump nubbin looking things, and they're radial polyps, okay? We also have them growing as digitate, where you get these small vertical upward facing branches over and over again, and your large table corals. I think everyone knows what we mean when I say table coral. And those are sort of the main growth forms which Acropora grow into, okay? They are ecologically very important. They provide a lot of habitat for small fishes and nurseries for juvenile. And they're also very important for recovering quite quickly after events of stress, such as a storm or a cyclone or a bleaching event or something like this. However, even though they're fast growing and they can grow up to 10 centimeters per year, they are also more susceptible to threats, unfortunately. And this is because, well, they're investing a lot of time into growing quite quickly. They also, as you can see, don't have the densest of skeletons, okay? So some of our larger, more bouldering corals, they are a lot slower to grow, but they are taking a lot more time spending the energy, putting the energy into creating calcium carbonate and extracting the, the components they need out of the water column in order to do that. So they're playing these very different roles. But these guys are really important for providing structurally complex habitat because nursery grounds are very, very important in the marine world. I'm just going to see if this was someone. Okay, beautiful. Imogen, I'm going to send you the taxonomy exam after this. Got your message. So let's very quickly cover the acroporas that exist in Caribbean waters. Now, I don't know, give me a thumbs up or a hands up or something if you've heard of just the state of corals in the Caribbean. Can anyone, does anyone know? Sort of a little bit, maybe? Brad, yes, okay. There's a couple of us. So the Caribbean is very famous because, unfortunately, it has a lot of coral diseases. This region has been hammered by coral diseases and other stresses to their corals for the last couple of decades. It used to be incredibly famous for its vast seascapes of acroporid corals. And then unfortunately, it was hit by a coral disease called white band disease, which had affected our Acropora palmata and our Acropora cervicornis corals. And they were also the two more abundant acroporids there. And they pretty much wiped through the populations, and in some places brought down the coral coverage by 99%. So they essentially nearly lost everything. So there's a lot of restoration projects and research, et cetera, being done, especially on these corals right here. And if you guys are interested in the literature and look it up in your own time, you'll see, you know, Acropora cervicornis, Acropora palmata in particular being mentioned a lot. So these are kind of, you know, very important species. So yeah, there are only 3 species out of the over 160 acroporas that exist globally. Again, mostly in the Indo-Pacific. This is the one that a lot of people refer to as the staghorn. So when people say staghorn coral, this is the growth form that they're sort of referring to. I don't think we have this sort of growth form quite in that degree in the Indo-Pacific, okay? Again, guys, so you guys probably noticed that I just mentioned the fact that these are different growth forms. This is what they grow as. When it comes to coral genera, it's quite variable. Each coral species grows a particular way. So a coral species will be branching, or it'll be plating, or it'll be bouldering, okay? For the genus level, because we're obviously looking at particular genera and they might have a variety of species, I'm just giving you the ones that they generally grow as quite frequently. But do just keep that in mind, that a particular species, you know, Acropora cervicornis grows branching, and that's the way it grows, and that's that. Just sort of a good thing to keep in mind. Okay, then we've got Acropora, which for me, I never had a mnemonic device or a way to remember Acropora. It was just mentioned so frequently and drilled into our heads and was all over the scientific literature that I just ended up remembering it. The way I remembered Acropora was that, like, it's like the very skinny, you know, brittle version of Acropora. They are in the same family. They do have these sort of nearly like rounded down axial polyps. They're not technically quite axial polyps. And I just remember it as like anorexic Acropora, which again, don't take these names seriously, but just by like word association, it's a pretty easy way to remember this coral genus. So these ones grow as branching acorran bows. They are very, very brittle, offering similar roles. They do make nice little nursery grounds for things to hide in, but again, particularly susceptible to, say, storm surge, waves, cyclones, and so on forth. The branches are very thin, 3 to 20 millimeters wide. So 3 millimeters sometimes is the entire thickness of the branch. So very small colonies. They've got blunt tips and very smooth sinostrum. So remember, that just signifies the space in between the polyps. And especially on this photo here, you can tell that it's very smooth in the middle there. Cool. So then we've got Isopora. Again, an Acroporidae. The way I remember this is that it does have technically some axial polyps, but they're like barely visible, or in some species they're just not there. So if you can think of Isopora being our Acropora, but its axial polyps had to go into isolation and go away. So it doesn't have those obvious tip ones, you know, at the top of each more hill here. These guys generally grow as encrusting, which is literally just when they're covering a surface. They grow in these submassive, which is quite a lumpy sort of growth form that can get quite large. Our lamina, which is sort of like our plating coral, rather thick plates too, or very thick branches. All right. Well, someone's getting back into the room. There we go, Brad again. Awesome. So again, very little or no axial polyps, okay? But the polyps are very similar to other Acroporidae, and by that I do mean our Acropora. So just keep that one in mind there. Then we've got Astrophora, okay? Still Acroporidae. Astrophora is such a weird coral because it grows in often these sort of very large, massive growth forms, as we can see on the bottom right there. And usually the general rule is when you get these sort of large, massive growth forms, they're usually pretty resilient to things such as bleaching, for example. Whereas for some reason in Astrophora, it seems to be the first one to get stressed out in bleaching. I know from my days in the field, when we saw a little bit of bleaching on Astrophora, we thought, oh, hadn't noticed it yet, but the temperature seems to be rising. And, you know, maybe a few weeks or a month later, some of the other corals might be bleaching too. Again, if we're having a heat year. And somehow it'd usually be the last to recover as well. But clearly this coral has, you know, got better susceptibility to like sustaining itself in times of stress, because, you know, they often did recover, but they would just take longer and sort of be the first to express those first signs as well. So a very interesting genus. It's sort of not usually the general rule, but again, science, nature does its own things, and there are exceptions to every rule. The top, very thin tissue, and it's a very pebbled surface. Corallites look like they're little domes covered in spinules. Spinules is a fancy name for saying bumps of skeleton. And we're going to see throughout this lecture that there are a few scientific ways to say bumps in skeletons, but spinules is the one we use to describe Astrophora, but it's bumpy. I think that this coral looks like, and I hope you guys see what I mean here, looks like someone took a bucket of googly eyes, stuck their hands in it and sprinkled it over the floor, and they've just landed all over the place. They're kind of facing all different ways, and all the little googly eye bits are facing different ways too, okay? I think the easiest way to remember this, and again, these mnemonic devices are sometimes a bit of a stretch, but just, you know, humor me. Acropora, I feel like looks like little asteroids hit this coral colony and cause these crevices, these big bumps in the central parts of the corallites. Because look at this, they're so dug out in the middle, and that's very typical of Astrophora. Astrophora, asteroids hit it. You guys are probably thinking, thank you, Elle, now I remember this coral so easily. Wonderful, you're welcome, guys. Anyway, so this one goes, I'm getting thumbs up from Trevor. Thank you, thank you. The rest of you I can't see. So in my head, you're smiling and going, yes, that was wonderful. Thank you. I go, no worries. Anyways, so these guys grow as massive or laminar and tiered. So that's sort of what we're talking about here. These sorts of like different levels of little shelving nearly is what it looks like here. All right? Wonderful. So then we've also got Montipora. Note everyone that we've got the little star next to it right here. Okay, so what that means is that I think this is of particular importance, and I think you should put it at the top of your priority list for remembering this coral genera out of, you know, I mean, remember all of them if you can, by all means, but this was one that's quite relevant to the Indo-Pacific and also mentioned in a lot of scientific literature. So it's a good one to remember. They have very small polyps. They grip together between skeletal bumps called monticules. So here is a bump name number 2 being, monticules. Montipora, monticules. These guys grow as encrusting, laminar, submassive, branching sometimes depending on the species. The polyps are small, just like a genus called Parietes, which we're going to look at in a moment. And some people say, that makes them quite similar. But actually, I don't really agree with that. Montipora has this very textured surface from all the monticules. And this is what I'm talking about here. All these little sort of spiky bits in the middle, or fluffy bits, those are the actual tentacles of each polyp. And again, they're nested in between all of these bumps called monticules. So Montipora has monticules, you might say has lots of little mountains across the landscape of this coral, and all the little polyps are just dangling, walking along between there. Must look like a bit of a mountainous landscape to them, or a monticulous landscape to them. All right, here's another few examples, just showing you guys how they can grow. So this is actually a different genus called Parietes, the one I just mentioned, very small polyps. These guys here, this is all Montipora. And here again, we can see it's sort of taking a laminar growth form. Looks like very nice, delicate little shells. This is a cool one called Montipora Varicosa, where the bumps are like on steroids. Those monticules are quite large. And often, even though it may not be obvious to us, unless, you know, you speak a language that, you know, has more Latin origin, like maybe like Italians or something like that, often the, you know, Latin taxonomy names of animals will quite literally just describe them. So in this case, Montipora is the genus name and Varicosa is the species name. And Varicosa, directly translated, pretty much just means warty, like bumpy, you know, like a bumpy wart that you get on your skin. And that's sort of just simply what it's describing right here. These very large wart-like mounds over the colony surface. And there are many other animals that do this too, like in the slug world, in sea slugs, a lot of the species names might be lineata, like Hypsilodorus lineata. And it's a Hypsilodorus sea slug, that's a genus that has lines running down its back. Lineata meaning, you know, lines or stripes, things like that. So even though it's, you know, unfortunately not really obvious, at least for us English speakers, it's actually really practical names that most of them have been given. So it's still really cool to think about, even though it's not maybe many of our native languages. So then we've also got Pavona. Note here, we've got another star next to the name, so a high priority to remember. So we've swapped over now from the Acroporidae to the Agoristhidae. So this is a really nice family. Has that thamnasteroid colony formation, which we'll show just in a moment on the next slide, a little bit more up close. The plates differ in orientation depending on their depth, which is so cool. So not that we'd ever just move a coral for the sake of it, but a single species of Pavona, you know, growing in the shallows that has sort of planting orientation, if it's very shallow, it might grow quite vertically, like the one pictured in the bottom right here. And that is because it's in the shallows. There's plenty of sunlight, you know, the water isn't getting in the way. Maybe the conditions are very clear, but they don't want to get too much sunlight. Maybe they don't want to overheat, which is why they're growing so vertically. If this colony were to be pushed or were to be somehow, you know, transferred to a deeper area, maybe part of the ocean that has got quite turbid, unclear water, or it's just deeper so that less sunlight is penetrating, what will happen is that those plates will start to grow more horizontally to pretty much expand the surface in which they can have access to the sun. So pretty much like, you know, shifting their solar panels depending on their location so they can best utilize their environment. And if that's not cool, then I don't know what is. But Pavona, very, very cool coral. It can be a little bit of a fast recoverer. It recovers fast after periods of stress. Where I was working, we didn't do much transfer of Pavona. When we would do coral restoration, we would work with naturally occurring coral fragments, say that got broken off during storm surge. We would never frag or break corals because we already had naturally high diversity. Again, in the Indo-Pacific, this is quite common. But we would do collections of naturally occurring fragments, and we would sometimes avoid certain genera because within that area they were in high abundance. You know, we'd prefer to spend our time working on corals if those fragments were available to us that did not have such high abundances. And, you know, if we were able to save a fragment of one of them that was struggling, it would be more sort of worth our while to use one that did have less representation on that reef than say something like Pavona in our situation. But again, a very good one to know. They grow as encrusting, growing over rocks, for example, foliose, what we're seeing here, and sometimes even submassive when they're growing a lot slower there. And good thing to know is that they have polyps on both sides of these plates. Obviously, these are 2D photos, so I can't show you both sides, but imagine you're looking at the other side and they look the same on both sides. All right, so here's another photo. These are two photos of the species called Pavona explanulata. That's the submassive one we were just talking about. And you can really see here, especially the lack of walls, the scepter just continuing on from polyp to polyp. Okay, now if you guys want to unmute yourself, if you want to pin up your hand, that's fine. But can anyone see anything weird about this photo on the right? Trev, what are you thumbsing upping? Do you see something weird?
Disease? Not sure.
Trevor says disease. Are you looking at this area here?
Yeah.
That's a really good observation. Does anyone, before I tell you guys what the deal is, does anyone want to make any comments or observations about that? If you know what the answer is, maybe keep it to yourself.
I know what the answer is, but because I've done this before.
Oh, that's
so
right. Oh,
myself.
It's a good one, isn't it, Zakia?
Yeah, it really is. Yeah.
It really is. Okay, so Trevor was totally on the right track. Well, okay, he's noticed that something is, you know, not quite right with the coral. That's the right track. It's not disease, though. This, my friends, is a sea slug and its egg trail. So we have got 2 sea slugs here from the genus Fastilla, and some of our team members at Conservation Diver described this genus or this species to science. So this here is a sea slug, and this is another sea slug. So there's actually 2 here, and these, you know, this sort of white yellow trail is a series of egg ribbons. They've just done the dirty and they're laying all their, you know, their fertilized eggs down so that they can, you know, develop and then hatch. So that's awesome. This was referred to as Fastilla species 3 for quite a few years. And then our buddies at Conservation Diver, one's called Spencer Arnold, one's called Rahul Mehrotra. They, you know, together with a couple of other people, actually ended up describing these guys getting samples, genetically testing, etc. And now it's called Fastilla V. And these guys are awesome. So their whole life motto, sorry, I know this is a coral lecture, but I'm just going to quickly do it anyways. They, you know, so their whole life thing is that they've got this coral, they look just like it, they eat it, they mate on it, they lay their eggs on it, and then they die there, and they just hope that nothing notices and eats them in the meantime. That's like their approach to life. And I think that's just so cool. They even will eat the coral and utilize the zooxanthellae in that coral. So for those of us who it's been a while for, corals are animals. They've got polyps and mounds. You can see them around here and here and here. And they've got a symbiotic relationship with a symbiodinium called zooxanthellae, which are little plants essentially. And that's what gives the corals their color. So these slugs eat the coral tissue and then keep intact the photosynthesizing mechanism and incorporate it into their mantles, because they're gastropods, and then they can even photosynthesize. Oh, that's so cool. Anyways, I just quickly want to show you guys 2 more. This is my photo. This is a Fastilla briarium, and this is a Fastilla melanobranchia. Sorry, I forgot the name there. So they look like different corals. This one, it's quite easy to see because these tentacles of the slug look just a few shades lighter than the tentacles of the coral here. But again, it just blends in. And here's another one. This one's not my photo. Where they've got the coloration of the coral behind it, and you can see the mortality that's being caused there. Anyway, back to corals. Just wanted to show you guys some cool other stuff as well. So let's move on to Dendrophyllidae as a coral family. Does anyone know any interesting facts about Dendrophyllidae? Oh, wait, actually, I just realized it says it on the slide. So I'll just continue. So the Dendrophyllidae, not every single one of them, but a lot of them are azooxanthellae. So that symbiotic relationship we're just talking about with the zooxanthellae doesn't exist for all the corals. So they are azooxanthellae, which means they do not photosynthesize because they haven't got that relationship with the plants. Okay, Tubastria are generally yellow or dark green. If their tentacles are contracted, like in these photos here, or not quite down the bottom, that means that they might be feeding or they might not be trying to feed at that point in time for some reason. And that's just the natural pigmentation, you know, of the proteins within their tissues. So they don't bleach due to heat, for example, like corals that are zooxanthellae do, okay? When the water gets too warm, most zooxanthellae corals might expel the zooxanthellae because the conditions are no longer right for them to continue photosynthesizing. But these guys don't have that reliance, which means that they can absolutely take advantage of conditions and locations that other corals cannot. Can anyone tell me where a coral that doesn't rely on sunlight might be found compared to a coral that does rely on sunlight?
underhangs of the reef.
Yeah, underhangs on the reef, exactly, but also just deeper in general. So where I've done a lot of my work, it's often the sort of standard coral genera that we see all over the place, they start to thin out and, you know, are less and less frequent after 15 meters or so, maybe 20 meters. Whereas these guys will start to become more abundant after those areas because there's all this available hard substrate that these other corals really struggle to make the use of because they can't photosynthesize, and that's where they get most of their energy from. These guys are like, we don't need the sun, we eat ourselves. We're heterotrophic, woo! And they can just live wherever they want to pretty much. Of course, there are other conditions that need to be appropriate, such as pH and so on, but they can make do without that light, which is really, really cool. So they've got a fascioloid colony formation. So again, you know, the polyps are very separated and tubular is the easiest way. And you get them quite branching and corymbos as well. And I think Tubastria is a very easy genus to remember because Tubastria looks like a series of tubes. Easy one, right? It's not as essential as, say, the Acropora in terms of the frequency of its mention, but I hope you guys remember this one because I think it's a pretty easy one to remember and also really pretty. Sometimes they're called, or often they're nicknamed, the sun corals. And you kind of, you know, when you see them with their tentacles extended, you really do understand why. Awesome. So then we've got another Dendrophyllidae, which is called Turbonaria. So these guys have got quite large spaces between the polyps. Again, scientific word for this is a synastrum. So this space in between where all the polyps are, pretty easy. So smooth-sided tubular corallites, which is talking about literally the sides of the corallites. So if you guys can see where my cursor is, those sides are very smooth up until you get to the tentacles, in which then it's, you know, tentacles galore. We don't have any costae. Remember, the septa come up from the mouth to the wall, and from the wall down would be where the costae are, but not all corals have them. And this is a coral that doesn't. Okay, septa would be there underneath the tissue, but they're so tentacly that you can't really see them, at least in these photos here. We typically get them encrusting in foliose, sort of vertical thin structure, laminar and massive sort of growth forms as well. Interestingly enough, I'm in Perth at the moment, and down south of here, even a few hours down south, I've seen massive colonies of Turbonaria and it blew my mind. So, so cool. I didn't think they were just existing in such cold waters. Sorry, just knocked my computer. But yeah, really, really great. Some people say they look like Astrophora. And when we're looking at a colony like this, for example, from a bit of a distance, remember Astrophora is the one that looks like little asteroids hit it. Very sort of bumpy, googly eyes being spread all over the place. That kind of bumpy nature from a distance can look a little bit like Turbonaria too. But we get up close, we can see that Turbonaria has got these daytime extended tentacles. They're also azizanthelate. Our Astrophora have got the little pebbled appearance to them, all these spiky little bumps called spicules as well. So those are the main differences there. So pretty good one. Good one to know. Awesome. So then we've got Diploastria, which is our first monospecific coral genus of the talk, I think. Yeah, no, it is. And it's called Diploastria Heliopora. Monospecific means that within the genus, there's only one species. So if there's a single species, I implore all of you guys to remember, I would say it's this one. So then you can be like snorkeling with your friends and you see this coral and you go, oh, Diploastria Heliopora, friend, that's what this is. And they'll say, holy moly, you know your coral species. That's so cool. And you'll just be like, yeah, I am what I am. I know my coral taxonomy. What can I say? I know all the species, even if you don't. But you can learn this one. So Diploastria Heliopora, single species. It's got large polyps ranging from 8 to 10 millimeters across, so maybe nearly a centimeter across. Usually it's kind of like low cones, like a cone that's sort of been pushed inwards. And its septa form these radiating sort of ridges, as we can kind of see coming up around here. And they eventually run down the walls into the costae, okay? And technically every coral does start as encrusting when it's just a few polyps large, but these are the corals, especially, it's just in the Indo-Pacific. This one doesn't exist in the Caribbean. Often forms these sort of huge dome-like structures, and they're very impressive. What, large Diploastria Helioporas can signify on a coral reef ecosystem is that, actually what they could signify if, say, you're diving in the area and there's very little in terms of coral coverage, but these guys are existing, you can think to yourself, okay, this area clearly does support coral life because this coral is living here. It's very slow growing, so this area has supported coral life for a long time. But if there's no coral anywhere else, or maybe you see the rubble leftovers of coral, what you can think, or what you can sort of conclude from that, is that there may have been some rather recent disturbances. So Diploastria are very good at recovering. They grow so slow, and they put a lot of energy investment into reserves. Whereas our other corals, like maybe our Montiporas and our Acroporas, they were both from the Acroporidae family that we were talking about earlier. They were more fast growing, if you guys remember. And even though they were fast growing, and that's all well and good, they're also more susceptible to stress because they weren't putting all of this energy into reserves and, lipid stores essentially. So they're also more susceptible to bleaching, to cyclones, diseases, etc. So presence of maybe rubble patches and lots of Diploastria says there's been some stress here, but clearly this is an area that can support coral life. And it can give you a bit of an idea about the timeline as well. Let's just look at some cool Diploastria photos. So these are provided by our good friend and founder, Chad Scott. And he sent me this photo once, and I was like, Chad, it looks like someone took a blowtorch to this Diploastria and a polyp is just like melting off the side. And he said it's budding off. And I thought, wow, I didn't know that was a thing, but here we are. So isn't that just crazy, everyone? Second photo on our right, we have got competition within species, but different individuals, okay? If this was the same, these are two Diploastria Helioporas, we can tell that they've got different clades of zooxanthellae, species of zooxanthellae within their tissue. So we've got this sort of different color happening here, and they're competing for space. Coral reef ecosystems are such a huge competition for space, like all the time, for that hard, valuable substrate, that rock or dead coral that we were talking about a little bit earlier. And when corals are growing and they meet, it's like fight to the death. And you get these really beautiful, interesting patterns forming, along the edges. One might be winning a little bit over here, a bit further down, the other one might be winning, but you can see that there's a little bit of, paleness and, it's nearly like a little bit of bleaching. That's just sort of stress of the colonies actually fighting each other, you can say, but really cool. And of course, this is just interesting because it's a war between two of the same species, which I guess is what we do too. Anyhow, there you go. Very cool Diploastria and all of its splendor. So moving on to different family, that being the Euphyllidae. So first, let's look at Euphyllia. Now, some of you guys might be thinking to yourself, that's not Euphyllia. I remember it being this other looking coral. Remember how I said earlier that as like taxonomy and genomic sequencing and DNA is technology is advancing, that we're pretty much getting more correct in terms of our taxonomy than we used to be. So this is one of those families in genera where there's been a little bit of shifting and we realize, oops, we did something a bit wrong before, we're going to fix it up. So Euphyllidae is pretty characteristic by having these beautiful long tentacles, and the next few slides and genera we're looking at are all going to have these sorts of characteristics. But yeah, we'll dive into it. So Euphyllia as a genus here has 4 species. The corallites are 2 to 3 centimeters. And you guys might be thinking to yourself, where's the corallites in this photo right here? And I can tell you, I don't know, they're under there somewhere. But we can't tell because they've got these wonderfully long daytime extended tentacles. The ends of the tentacles have these kind of really cool, lighter colored knob-like ends. That's pretty characteristic of all the tentacles within the Euphyllidae family, at least all the ones that we're going to be looking at in these lectures. okay? And the colonids are fascioloid. That was the colony formation that we were talking about before, separate corallites, but we actually can't see unless it was dead and all that was left was the skeleton, all right? So let's look at the next Euphyllidae, which is now called Fimbriophilia. Some of you guys have been to the program, New Haven Reef Conservation Program, which is the first conservation diver centre that existed. And it used to be that this was called Euphyllia. Sorry, it's been reclassified. So now it's called Fimbriophilia, okay? And it's got the daytime extended tentacles, these often, you know, kind of jelly bean or hammer-like corals. This is often nicknamed the hammer coral, but it's got that kind of glow and light tip to it, just like the Euphyllia tips do there. The skeleton is kind of similar composition to porcelain. It's got a flabellate colony formation. And that's what I was saying before, earlier on in the lecture, about these, it looks like there are big gaps, like big chunks missing out of the colony, and there isn't. That's just the way it grows. When the tentacles are out, again, you can't really tell. Whereas when they are retracted here, if they're feeding, if there's been a disturbance, that's when you can actually see it, okay? And we get them growing these sort of massive and submassive colonies. And I forgot, guys, Euphyllia. One of my friends, Kate, one of the other instructors at Conservation Diver, she said, always remember Euphyllia because it's just so pretty and, you know, you have these long tentacles and I love it and I feel euphoric whenever I look at it. And I go, that's a good mnemonic device. So she had that little word association just because it happened to be one of her personal favorites. Whether it's yours or not, you can choose if you want to, you know, remember Euphyllia, you know, with the word association, with the euphoric as well. Up to you. Now, Galaxia, which is just one of the, this is such a cool coral, guys. I really love it. We're still part of the Euphyllidae. We've still got daytime extended tentacles, but we've also got these blade-like septa. And it's a little bit hard sometimes to tell the difference between the septa and the tentacles. So if you'd like to glance over to the right side of the page, we can see these bits that are kind of sticking up a little bit more vertically. These are the blades, the septa, which was that, you know, structural like part of the skeleton, which goes from the mouth to the wall. In this case, it doesn't do that. It's just like, septa, attack. And it just protrudes out of the colony or out of the corallite completely. And they are very vertical. And the fact that they're covered in tissue makes them look very unalarming. But, you know, if there's a coral I would not recommend bumping into, not that we'd ever bump into corals, it would be this one, because I assure you it would leave a mark. Anyhow, then we've got the beautiful tentacles out the sides here, okay? And much like, again, the other tentacles of the Euphyllidae, at the very tips of each of these tentacles, you get that little extra light coloration, okay? And especially because it's very short tentacles, they vary, you know, they're moving very quickly when there is a bit of a current. And I always sort of look like, you know, when you couple those sort of things moving around, gushing about, with the fact that these corallites are just so star-like or flower-like in shape, plus the glistening of these, the tips of all of these tentacles, I always thought it looked like a little glistening, you know, starry night, which leads us into our next mnemonic device of Galaxia, you know, being like the galaxy, a night star, a nighttime sky, you know, glistening back at you with all these rushing around tentacles, okay? So hopefully you guys can appreciate this coral as much as I do. Ethan, which is on this call somewhere, she just has the most wonderful photos of these corals and it makes me smile every time I see it on her Instagram, Coral Reef Beauty. I think she's still around somewhere. I actually can't see everyone on the list to the right. I hope you're still there, Ethan. Maybe. Anyhow, so it's a very distinctive coral colony. And moving on. So let's talk about some of the Fungidae family, otherwise known as the mushroom corals. Yay! The mushroom corals are very cool because they are, generally speaking, unattached individuals. You know, when we learn about the basics of corals, we usually learn, they're colonial and they're sessile, attached to a hard substrate on the reef floor, and then they grow into these magnificent structures. And the Fungidae family of corals just said, nope, we're not going to do any of that. And generally speaking, well, about half the time they're actually solitary. So they are a single polyp, okay? And a lot of the time they're also not attached. So the two large generalizations we often say about corals, these guys just didn't do that. So let's look at their structures here. So these are a few different variety of fungids that we're going to be looking at. Here is one here. Up the top, so you can see a bit of a slit there. That's its mouth. That's the center of the polyp. To our right, we have a juvenile. All of them are attached when they're juveniles, and then they'll detach. Kind of looks like they've got a little belly button scar on the bottom sometimes, especially when they're still quite young. But you can see the mouth quite easily in this little one there. And the septa, just like on all the other corals we've been looking at, these guys have got septa. I used to be a little bit confused about the septa. You know, the septa are here, sure, but where are the costae? I nearly thought that it was like coming up from the mouth, you had a little bit of septa and then a lot of costae coming down the side. So costae, septa, but that's actually not true. So what mushroom corals have is septa, and then on the underside, if we go to this top photo, then it curves around and becomes the costae below. So you see these lines. This is very similar structures to those colonial corals that we were talking about before, but it's just one side is the top with the septa, the bottom side has the costae. Okay, we've got a lot of different types of septa. This is how we help identify some of the different mushroom corals. They can be quite saw-like, they can be a bit smoother, but we'll get to it as we go. And they do have tentacles only on the top half, which you can see in this photo just there. So let's jump into it. So first we've got the Fungia genus. So Fungia used to have over like 100 species in it. Don't quote me on that. I don't know if it was 100, but it was a lot of species in it. And then they did all of this, you know, testing, and they worked out that we were just quite wrong with that it wasn't all Fungia, you know, genus with a few subgenera. It's actually Fungia is a genus that has got 2 species in it. I thought up until yesterday that I actually only had 1 species, but I've now learned that there's 2. Both of them only exist in the Indo-Pacific. Fungia fungides exist pretty much all over the Indo-Pacific, and Fungia pushiani exists just on the west of the Indian Ocean. So depending on where you are, you can forget the species name if you want to. Fungia fungides is the much more frequenter of the Indo-Pacific in general. Just as a side note, I think this family is interesting when it comes to Fungia fungides. And when you say it, its family name is Fungidae, genus name is Fungia, species name is Fungides. And I just feel like that wasn't so creative, and it's kind of a funny family name to species name combination. So anyways, so these guys are solitary, and they grow up into these discs that end up being around 30 centimeters across at maximum. They have got rather ragged septa. They don't have these sorts of saw-like septa that we just looked at before, but they are kind of nearly quite sharp in nature. Like if it looks like if you're going to run your finger along that, you might, you know, cut it a little bit. This is how they look like when they're juveniles. You can see they've encrusted onto a rock here, and there's quite a few juvenile individuals budding off there, okay? And of course, this is what they look like in their adult form. Then we've also got Dendrophyllidae. So of all the mushroom corals, I think these 2 are probably the easiest to mix up. They've even got similar names, and some of the characteristics do overlap, okay? They both grow to be around that 30 centimeter mark in terms of the diameter. They're both solitary and free living and circular. Dendrophyllidae generally have a thick disc. I can find they can be a little bit thicker than the Fungidae, and they can have a central arch. So it kind of goes up a little bit in the middle, just as we can actually see in here. This is the Dendrophyllidae as well. This is actually a very nice example of that central arch, but all of them are not going to have it so distinctly either. But the septal teeth are very saw-like, as you can sort of see across some of these photos, like in this part here, but we can see in this black and white image that it kind of looks like an old rusty saw that got thrown in the garage, and you pull it out a few years later and it's a bit chipped and battered, but it's still, you know, an old saw blade. That's sort of what these guys look like. Additionally, the costae spines are large, especially on the edges. They're very, very tall and spiky, and they get smaller in the middle, much more distinctly so than the fungia. And me personally, that's how I tell them apart. I think the costae spines are the easiest place to spot it. Cool. So this is obviously not our work right here. This is from Hope Sama, an awesome guy when it comes to everything mushroom corals. I'd like you guys to just, you can look at this if you want to, but take a focus to the right-hand side. And we're going to look at the Fungia septa versus the Dendrophyllidae septa. You can sort of see the difference there. And you can look at my very artistic interpretation down the bottom here. Fungia are quite uniform, and they're quite, uniformly ragged and even all the way along, as you guys can see here. And the Dendrophyllidae are sort of all over the place. Again, looking kind of like rusty old saw that might be a little bit broken, as we can see along here. All right, so second monospecific genus of the lesson, Herpalitha. So Herpalitha sounds like herpy. You can use your imagination to work out a mnemonic device for that if you so wish. I did, and that's all I'll say. Anyhow, so the species name is Limax. So Herpalitha limax here. It's quite a cool, unique mushroom coral. It's very long and extended, and the edges have a bit more of a point to them, compared to our Dendrophyllidae and Fungia that we just looked at. They are very nice and circular. Not all of them are circular. We'll look at some other ones in a moment, but this is probably, this is certainly the longest, skinniest, and pointiest of this group of animals. We have got a long central fossa, which I just realized I don't think I explained before. Fossa on a mushroom coral is the same way, it's describing a mouth, essentially. Interestingly enough, Herpalitha is colonial. So it's not a single polyp like our Dendrophyllidae and Fungias. So we have a long central mouth fossa, as we can see on this photo up the top. And all these little white specks that we can see are actually tentacles. And if there's tentacles, that means there's a little mouth there that they're feeding. So they've got these nice septa that start at the fossa, starting at the central fossa, and they don't usually make it all the way down the edge without meeting at least one or two other mouths, okay? They're also quite hollow below. So if you were to flip this over, you would see it's nearly like it would make a lovely long bowl that you could like drink water from or store something in. I don't have any photos of that right yet. By the way, if you guys want any more specific examples about any of these corals, I've got a whole personal database on my computer. I can show you guys after personally, whatever you guys like, by the way. So again, elongated in shape, they can branch, which is not very typical for mushroom coral, but occasionally they'll do it and they might make Y, T, and X shapes. Nature just breaks the rules sometimes, and that's what these guys do. Wonderful. So then we've got Sandalolitha. And I remember when I first came to the program as a staff member, that was at New Haven Reef Conservation Program based in Thailand. We'd just gone from referring to all of the mushroom corals as Fungia to referring to them all as their now more correct names. And I remember one of the interns at the time, Tia, she's so lovely, she rocked up to me and said, Elle, I have the best mnemonic device for this coral. She said, and I said, all right, amazing, give it to me. She said, Sandalolitha sounds like sandal. And wearing this coral on your foot as a sandal would be so uncomfortable. I was just laughing so much. I was like, dude, to be honest, like if you put any coral on your foot, that's not going to be that nice, I don't think, for the coral or for your foot, They've got ragged septa and they're pointy and they've got stinging tentacles. But I thought, it's kind of funny, and I remembered it. So I think they kind of look somewhere between like a lumpy figure 8 and a witch's hat. They are also colonial. They do not have a central fossa like the Herpalitha. Okay, so they've got many, many different mouths, and they actually take a thamnasteroid colony formation. So if you remember that bifacial coral we were talking about earlier, called Pavona, which can grow vertical or more horizontal depending on its exposure to sunlight, this has got the same colony formation as that one. So the septa just continuing, there are no distinct walls there at all. The polyps are inclined towards colony edge. Pretty much what I mean by that is that the septa run towards the edge. They don't run parallel to the edge or anything like that. And the septa are also covered with uneven gradulations. So it's nearly like they're a little bit, bumpy or saw-like, as we can see in this photo right there. And there's 3 species in this genus. Is everyone doing good still? Any questions or problems understanding my accent or anything like that? Cool, thumbs up. Awesome, I'll just continue for now. So then we've got Tinactus, which I used to say Synactus, which is wrong, but also it's Latin. So there's not that many people who are going to go up and correct you, I suppose. We've got 3 species within this genus. It's got very nice, large, saw-like septal teeth, as we can see right here. And they're quite a large mushroom coral. They easily breach, the 30 or 40 centimetre mark, no worries. They are generally got a single mouth, but sometimes in this genus, I have noticed you might get a break and you might have 2, sometimes even 3 mouths there. And some of the other mushroom corals can do this too, that are more typically just got the 1 central long fossa, okay? Occasionally they branch, making that sort of X or Y shape, as we can see in this example in the far bottom right corner, sort of like is a Y shape here. They're elongated, and of course they're free living as well. Yeah, and I've got a bad mnemonic device to that one that I just forced myself to think up. You can refer to the PDF if you want it, but I'd more highly recommend you think of one yourself, which is better than mine. Okay, so now we're moving on to the Lobophilidae. We're done with the Fungidae family. If you guys hadn't noticed, we're going through all the families in alphabetical order. So Echinophilia has got 11 species, okay? They grow it. They grow in a submassive growth form, sometimes encrusting and also sometimes laminar, as we can see in this photo right here. The corallites are quite large, 5 to 10 millimeters. It's large when they get into that 10 millimeter size. They are quite untidy and coarse, and they've got large, uneven septa, again, going from the corallite mouth up into the wall. It's very bumpy, and they're also very toothed, as we can sort of see across all of these photos. I think Echinophilia, me, coming from Australia, we've got these animals called echidnas, which are kind of like the Australian versions of, hedgehogs. And because these are bumpy and spiky and small and round, I think that reminds me of an echidna slightly in my crazy stretch of the imagination. And I just made the little connection with echino and echidna. So Echinophilia is a little bit like an echidna, and that's what got me to remembering that one in the end, after I wrote down the word Echinophilia about 20 times or so, sort of glued it in there, all right? Then we've also got Lobophilia. So this is from the Lobophilidae family still. Lobophilia is pretty common around the Indo-Pacific. Often it's described as large brain corals. I'm sure you guys can understand why. Think of Lobophilia as a brain, your lobes in your brain have got, lobe in shape, I suppose is how we describe it. So it's kind of pretty easy to make that association to Lobophilia. Large flablate corallites, they break into cones that reach the center of the colony. So not that you would, but if you were to crack one of these colonies down the middle, you see like these sort of cone-like structures. The big kind of lobes here will just get thinner and thinner, like an ice cream cone. They've got very thick, fatty tissue. If you ever did see a dead one, you would notice that the septal teeth are very spiky and sharp, but you're not going to notice that unless the tissue's gone. And they do grow in these massive curved growth forms. So now we've got Diploastria, which is it's new. It's relatively new. You guys might notice down here that I've got a C plugged on this photo. So Diploastria is, within the last few years, new to what we call these sort of donut-like corals in the Indo-Pacific, okay? We used to call them Favia, and we also used to think that like a lot of these sort of circular, corallided, looks like little donut-like corals with favia in the Indo-Pacific, but we were wrong. Okay, it's sort of been reclassified. Now it only exists in the Caribbean. This is a favia fragum. This is not my photo. It's just source down the bottom there. They are found in the Caribbean, and it's been also realized that they are not actually at all related to the corals in the Indo-Pacific, even though their morphology is very similar. I mean, as you guys can see in these photos right here, I mean, they do have some quite clear characteristics that look quite like each other. So I used to love saying favia was my favorite donut, and that was how I remembered it, because it was a donut shaped coral. You can absolutely do that if you're in the Caribbean if you want to, but in the Indo-Pacific, please do remember that this is now called Diploastria. They grow as massive and submassive. They are usually quite nice circular donut shapes, but they have got this intratentacular budding. So occasionally you'll see these sort of figure 8 shapes when they're pinching off to become more polyps, or when they're growing more polyps, I should say. Okay, in this photo in particular, you can see the obvious scepter to costae area. It has not got a smooth synastrum. You can see that the costae run all the way down until they meet with those from the bordering corallites there. And additionally, the corallites, being the whole, skeletal part of where the polyp sits, protrude out slightly, as you can see here. So also, I forgot, we're in the Merulinidae now, which is another family name. Great. So then we've got Montipora. This is not in the Indo-Pacific. I just wanted to mention it because we used to, again, say that it was a coral in the Pacific. It's not, it's actually monospecific. It is Montipora cavernosa. It is one of the more commonly referred to corals of the Caribbean waters. So again, I wanted to include it, even though this is a predominantly Indo-Pacific based lecture, that this is just one of those general species of note, of significance, if you guys did want to remember this. Also quite donut shaped. I think it looks somewhere between our Diploastria and our Diploastria Heliopora, which is the first monospecific coral that we talked about in this lecture. This has also got extra tentacular budding, which makes it different to the favia fragum that is also found in the Caribbean, that also has very circular sort of corallites there. All right. And then we've got Acropora, also part of the Merulinidae, which has got irregular corallites, synastrum covered in fine spinules. So we've got spinules, we've got spicules, we've got monticules. Again, all just names describe bumpy skeleton. If you want, you can just say the skeleton is bumpy. That's totally fine. Acropora is the thing that we're aiming to remember today. They grow as encrusting, laminar, and submassive, okay? These ones can be a little bit confused for Astrophora, because if you remember from the very beginning, from the Acroporidae family, Acropora have also got that sort of scattered, irregular corallites, googly eyes scattered on the floor kind of look to it. But Astrophora has a very dug out appearance in the center, whereas these guys do not. They've just got these nice bumps. I also think these are a little bit echidna-like. Acropora is the other echidna coral. These little mounds that just tuck themselves in and they put out their bumpy, spiky back so that nothing is going to hurt them, okay? At least that's what echidnas do when they feel like they're being threatened. But again, irregular corallites, space in the middle, and that space is bumpy. Sweet. So I just want to pre-warn you guys that the next few genera are a little bit tricky, but it's going to be okay. We're going to get through it together. Don't feel like you're going to have to remember all these on the first go, because no one ever does, except for some people. And let's be honest, I don't like those people because this took me forever. We'll just jump straight into it. So Favai's got 20 species. It has irregularly shaped corallites. Some of them look like little squares, some of them are triangles, some of them are ovals. So remember, Favai's has got irregular corallites. We've got shared walls. They're not separate between corallite to corallite, like in these guys here. That's its own wall, that's its own wall. Whereas in this one, the walls are shared between all of them. The corallite's also very angular from the top of the wall to where the mouth sits. It's on this nice fine angle. Okay, so that's another way to remember it. It's got medium sort of clean cut septal teeth as well. They're quite neat and they grow as massive, submassive and encrusting. Sometimes we get these cool color differences between the walls and sort of the base of the polyps. This is not that much, but you sometimes get like greens in the middle and yellow on the wall. And for me, I always thought, wow, that's so pretty. Phovides is my favorite. And again, Phovides, favorite, similar words, even though nowadays I've got about 100 favorite corals. Anyhow. Now we're going to talk about Goniastria, 9 species, and it's got an excavated appearance, okay, quite dug out in appearance, like you can see in this Goniastria minata. It can weakly meander, so it nearly looks like it's trying to fully meander and become, like not puzzle, like a maze, but it's sort of not quite getting there, as we can see in these photos down here, okay? It's got irregular shaped corallites, like our Phovides, but they're much more dug out than our Phovides. This is probably the nicer example down here. The corallite's got neat septa, and what I want you guys to remember is that they've got paliform lobes. So if you remember from the beginning, skeletal structure, I said paliform lobes. A lot of corals don't have them, but when they do, they're quite useful to remember that they do have, because they're often the distinguishing factor. And this is going to be a distinguishing factor for us. So it's kind of like the secondary wall or crown just before you get to the mouth of the corallite. As you guys can sort of see here, you got the wall, it dips down, and then there's like this sort of secondary wall, and then you've got the mouth down there in the middle, in the bottom, okay? You can sort of see them along here as well. So paliform lobes are going along with Goniastria. Goniastria sounds like gonads. When we do coral spawning work, we would often work with Goniastria a lot because they're quite a resilient species. So it'd be sort of on our priorities list, and we'd play with their gonads. No, well, sure. Say that if it's going to help you at all, you just think of it like that, sure. These guys grow as massive, submassive, and branching as well in very specific examples. Deep breath. And then we've got Paragoniastria. So I've got Garoniastria, and then we've got Paragoniastria. So and one that I really quite like is called Paragoniastria australensis. It used to be chucked in with the Goniastria genus, and then scientists were like, no, that's wrong. Actually, it's got to go in their own genus called Paragoniastria. And so they did that. There are 3 species, and here's an example of all of them. One is full on meandering, baby. And look at that. It's like a crazy maze that is just inescapable. And then we've got some that are just sort of your typical, more circular, round corallites, as you can see here. Racelli, Deformis, and Australensis. That's my favorite one. They grow as submassive and encrusting. They are meandroid or steroid. They've got quite uniform septa, especially the Australensis. Look at those. Look how neat they are. I love it. They've also got paliform lobes present, but I think they're more present in the Deformis and the Racelli, just personally speaking, from me looking for them, amongst corals and so on and forth. They're quite obvious, especially like where I'm putting my cursor right now. There, it's like, boom, here's my mouth crown, here's my paliform lobe. And there we go. Also, the walls and valley floors can contrast in color, just like in literally all these photos. You got like brownish to the green. You got the kind of blue to the blue-green to the white. We've got the green to the pink. So they're aesthetic, at least, even though they're a tricky genus. And then we've got Platygyra. I know this looks a bit different. This looks a bit similar to Paragoniastria australensis, which is over here, but it's okay. On the tops of the walls, Platygyra are a bit more like this. So you got your valleys, and you got walls, and they're quite flat at the surface. On the Paragoniastria, they kind of like turn into this nice, like sort of like rounded bit. I'm going to go back for a moment. As you can see here, kind of like a nice point. Platygyra is not quite like that. They've also got shared walls, of course. They are fully meandroid. Valleys and ridges also can have the contrasting colours. And sure, they can be confused a little bit with the Goniastria and the Paragoniastria, but these guys definitely no paliform lobe, like there isn't even a hint of them. In some of the Paragoniastria australensis, you're looking down the valley and you're thinking, oh, it sort of is, sort of isn't, I'm sort of not sure. Whereas these guys, you look inside of it and it is incredibly flat, okay, in those valleys. So honestly, I know it's still a little bit tricky, and time it gets better, but that's sort of like the characteristic that I go for every time. Okay, we can discuss later if you guys want to, but we'll move on to Hydnophora. We are sort of leaning towards the later end of the corals that we're discussing today. So I hope none of you guys are, you know, hating me too much at this point in time. But I just wanted to tell you guys as much as we could and make the most of this. So Hydnophora is still part of the Merillinidae family. Hydnophora, if you guys remember from the colony formation slide, like at the very beginning, there was this colony formation called Hydnophoroid. That's for this one. Other corals don't do it. This one does. And they were like, we got to think of another way to describe this skeletal bump. So they came up with the word hydnophores. Okay. So polyps of this coral are nested in between these hydnophores, and they've got these beautiful daytime extended tentacles, okay? I honestly, guys, to ID this, wouldn't bother even looking for the polyps. All you've got to look for are these large bumps, these hydnophores, and you're good to go. They can often look a little bit fuzzy, like in this colony on the bottom left-hand side. And I always just, you know, remember the word association by saying hydnophore has got its polyps hidden amongst the hydnophores. It's because like I usually can't see them because they're covering the tentacles, or the hydnophores are in the way, and they're dense anyways. But this is a very, very, very nice, beautiful colony for sure. Actually, probably not as common and frequent as the other ones we've been talking about today, but I feel like its beauty level was worth throwing it into the lecture. So if you guys have seen it in real life as well, I hope that you guys can appreciate that one there. So then we've got Merillina, which I think is the last of the Merillinidae for this family. It's a little bit, it's not confusing, but it's not in one way or any other really distinctive. So what we can remember, well, firstly, the growth forms are encrusting, laminar, foliose, and submassive, but it has got rather indistinct correlates. There's one here, here's another one, there's a couple like that. And they're in quite short valleys, which you can probably see demonstrated a bit more in this photo down here, that fan towards the colony margin. So they're always heading outwards, as we can see here, as we can see along here, and as we can see just all around the entire edge. It's never going to be running, those valleys are never running parallel to the edge of the to the edge of the coral. They're always running towards it. And for me, if you look at the tops at least, kind of looks like uneven zippers, okay? You don't get a contrast in between the valleys and the valley floor like we do in some of those other corals in the Merillinidae family that we were just looking at. Phew. Acropora. So anyone that's been to the New Haven Reef Conservation Program should know Acropora. I've put a star next to it. It is just so dandy at growing when there's space available. And it's even believed, and pretty much, yeah, it's got maybe an extra spawning season even in many places around the world. So it'll spawn sort of mid-year, but also towards the end of the year as well. Very special. It is also a brooding coral. So pretty much in the evolutionary timeline of corals and coral sex, these guys are a little bit more advanced. What they will do, instead of just synchronously expelling their eggs and sperm together and then being like, good luck, kids, we hope you fertilize, these guys will expel their eggs, and then other colonies will absorb the eggs and fertilize them with their sperm. Then they will make a little larvae and put some zooxanthellae into it, so that when they eventually shoot the larvae out, they've got a renewable energy source. They're like, hey, kid, we love you. Here's a solar panel. And that's actually how these guys spawn. So they are a little bit more advanced, which has got, gives them a lot more success in terms of having their offspring survive. But an implication of that is that it can lead to more genetic bottlenecking, especially depending on where the colonies are placed, where the spawning of the eggs is happening and absorption. You know, you can get a lot of genetic similarities happening there as well. But very, very cool in terms of their approach to spawning compared to a lot of the other older corals. Anyhow, Acropora, a very successful genus. The Acroporidae in general are very good. You've also got the Stylopora, the Seriatopora. Depending on your region, you might see those ones a lot. If you want more information on them, please let me know. But this one, I think I see most diving I've done around the Indo-Pacific. We've got these very blunt-looking branches that have this very fuzzy appearance pretty much all over. And they've got these very, very short, skinny tentacles that are mostly always out, give them that sort of fuzzy all-around appearance. They're immersed, the small coralites are immersed across the colony's surface between the varicae, which is also kind of like little bumps, which is not too obvious on these colonies here, but it can have a bit of a bumpy skeleton as well. They also provide that very intense structural complexity like our Acroporas did that we were talking about before. Again, providing cool habitat for damselfish, symbiotic crabs, and so on. They grow corymbos, which is the dense branching type of growth. Okay, we don't get them in nice long skinny branches like Acroporas. This one is very, very dense. And it is what we call a pioneer species. Does anyone know what I mean by a pioneer species at all? Sort of a term that probably isn't thrown around that much. Okay, I'll tell you. So, you know, in the literature, you sometimes do get corals being described as pioneers, and think of it as literally like, what do we mean when we say a person is a pioneer? Maybe, you know, they'll go out and explore the ocean and find a new island. Maybe, you know, that person might be described as a pioneer for finding more land. We can, you know, this is also what we're describing for corals. So I said earlier on that corals require hard substrate in order to grow, and they pretty much do, but some can do a bit better when the conditions aren't quite right. So say you've got a field of small rubbly patches or something like that, okay? For some corals that are very slow growing, if they happen to recruit onto a piece of unsecure rubble, they might get a polyp develop, they might, you know, asexually produce a few more, but if like some surge comes through and that piece of rubble turns over, it might be the end of its days and it might not do very well. Whereas a colony like Acropora can essentially like go and recruit and start asexually producing so quickly and invest a lot of energy into growth that even if with time that fragment rolls over and dies, the colony might be big enough that enough of it survives, it's still facing the light, it'll grow back over and grow back over. So even if there is a little bit of movement, it can actually, it's shown to do, actually outcompete the movement of its substrate. Occasionally, even breaching off into areas that were previously uncolonized by corals. They find a little bit of hard substrate. If it's not even 100% secure, it might just be enough for them because they really do invest a lot of energy into growth. So they can kind of outgrow the disturbance nearly. So a sick colony in terms of resilience and, you know, hard work and, yeah, tolerance to moving substrates, I suppose we can say. All right, guys. So parietes, we mentioned this one earlier, having lots of little small pores. It's got a star next to it. So I'd like you guys to put it at the top of your priorities list if you want. I think it's a pretty easy one to remember. Parietes, with the exception of parietes negrosensis, is generally speaking a submassive, funnily shaped coral colony with a lot of small polyps that you could just say look like pores. Think of us humans. We are funny shaped animals with dangly bits on our extremes and funny shaped faces. And if you look up close, our skin is covered in pores. So parietes is a porous animal thing. If if you want to remember it like that. I think it's one of the easy ones to remember. So the corallites are very small, 1 to 2 millimeters. We get them submassive, as is on these outer two images, and then branching, as we can see in the center here as well. Sometimes they produce a pink to purple pigment if they are feeling stressed. Some of you guys may have heard about fluorescing corals when they're under stress. Fluorescing exists amongst all corals all the time, but usually we can't see them without UV light. But sometimes under stressful conditions, even our weenie eyes can pick up on the fact that there is a stress response happening there. And they can use it to either absorb more light if they are deep, and sometimes they use it to reflect light more if they are too shallow and suffering from, say, stress heat. So really, really interesting there. And that's a very, very, very shallow explanation of it. But it is a stress response. Also note that there is a C plastered right here, which means Parietes as a genus also exists in Caribbean waters, okay? And is a contributing reef builder there. Sometimes you'll find that a lot of Christmas tree worms will exist on Parietes corals. Not everywhere you go. In the Gulf of Thailand, these Christmas tree worms love Parietes. I don't know why, but it's a nice little relationship they've got going there. It's even suggested that these little Christmas tree worms actually protect corals like Parietes from predation from crown-of-thorn starfish. They like tickle the bottom of the starfish, and the starfish are like, I don't want none of that. And then they'll go away. So they are shown that in instances where there are Christmas tree worms versus there not being, they are less likely to be predated on by crown-of-thorns than when there aren't Christmas tree worms. So how about that, guys? Beautiful. There's just a couple more examples of the different varieties, the branches, the plates, the kind of awkward submassive shapes here. All right, so then we've got Goniopora, which is Parietidae as well. We have got these lovely long daytime extended tentacles. Some people look at this, and I know this is a 2D picture, but if you see it live, it's sort of like all flowing about because it's got these lovely long tentacles. And a lot of people think that's a soft coral. It looks soft, and I totally get it. It does look like a soft coral. But if you kind of try and imagine what its base structure is, that isn't moving. There is indeed skeleton below. If you want to, you can give it a bit of a wave, and that, well, don't do it just for the fun of it. But if you're really unsure and you're doing a survey, you can give it a little bit of wave, and that disturbance will often cause the tentacles retract, as you can see they're doing in this photo here. Also, these guys at the tips of their little flower-like tentacles here have got 24 tentacles. You guys hopefully remember me saying earlier, Sclerotinia have got tentacles in multiples of 6. Octocorallia have got tentacles in, well, they have 8 tentacles all the time. These guys' tentacles in multiples of 6, and this one has 24. There's another genus called Heliopora, which looks, so similar to Goniopora. The difference is they have 12 tentacles pretty much, okay? But Goniopora probably a little bit more common, depending on your location. Here we can see an example where you really have both happening. So you can see the tentacles are long, extended here, and they are retracted here for some unknown reason. Cool, so let's look at one of the bubble corals, just because often people mention bubble corals, so I thought it's quite useful to include it here. We have Plerogyra from the Plerogyridae family. It's got 7 species. Tentacles are extended during the day. To be frank, I've never seen one with the tentacles retracted. I've tried before giving it a little bit of a wave, and it sort of seems like it tightens up a little bit in terms of these bubble tentacles, but otherwise not that much happens. Understandably confused for soft coral. But you can see there is some solid structure below that, you know, it's not wobbling like the whole structure of it. But if you remember Plerogyra, yeah, the bubble coral, even though it's bubbly and looks soft and lovely, is a hard coral. Just one that, again, people have asked, so I just threw it in there. But I won't go into too much detail about it. Unless you guys want more photos or anything later, just let me know. All right, so then we've got Pachyceres. You guys might think, what does Sclerotinia inserte sedis mean? Pretty much all that means is that the family is still being confirmed in terms of genetics by the experts that deal with Pachyceres and other corals similar to that. So it hasn't got like an exact confirmed where does this sit in terms of the taxonomic tree quite yet. So Pachyceres has got an interesting form. It is sort of meandering. You guys might remember before, it's meandering Pachyceres type. That's describing here. I remember when I had my first coral taxonomy class with conservation diver, and my co-worker, Powell, gave me this lecture, and a variation of this lecture. And I remember he got to this slide and he goes, oh, Pachyceres, elephants. And I was like, what, man? Yeah, I can see your face, Trevor. You're saying that to me right now. Like, what? I was like, hey, Powell, like, what are you talking about? He's like, elephants, their ears are wrinkly. I was like, yeah, cool. But like, I'm still not sure what you're talking about. And he goes, taxonomy, elephants, family name is Pachyderm. There you go. So the elephant family name is Pachyderm. Elephants have wrinkly skin to aid in heat loss. Pachyceres sounds like Pachyderm. Pachyceres is a genus of coral with folded, folded like growth form. So obviously this is where the connection is. And I thought, oh, wow. And now I've never forgotten the name of the elephant family or this genus of coral. So I hope you guys come out with the same outcome as I did. And I didn't know either of them though when I went into this. Anyway, so we've got small indistinct corallites. You don't even need to bother IDing them precisely because you just look for these wonderful valleys that run parallel to the colony's edge. There's nothing quite like it. It's a very, very distinct genus that has 6 species. It grows encrusting, laminar. It really does look like some big floppy elephant ears. Like he wasn't wrong. That's what it looks like, you know. This photograph to your left here, it's not vital or anything, but just because of the sheer like beautiful detail of the septa which run along these valleys, when you have little invertebrates boring into the actual skeleton, the skeleton reacts by, trying to grow over them. And it just, these phenomenal little patterns emerge. And I just think it's so cool. So I just wanted to include that photo there for you guys. Anyhow, Pachyceres, elephants. So guys, we have a little bit of a quiz. Here's Squidward from SpongeBob SquarePants, enthusiastically looking at some coral in his backyard or something. This isn't the exam that I can send to you guys after if you want to test yourself. This is just, I think, like 5 or 6 corals that I think it's 6 corals, don't quote me on that we've gone over today. You guys can unmute yourselves. You can go into the chat if you want to, but if you remember them or even remember something about them, just say it out loud and we can work it out together. So, what's this?
Diploastria.
Yes, wonderful. Diploastria. You nailed it on the first one, Anthony. So that's Diploastria Heliopora. Legend. Awesome. I should have written Heliopora there. I just wrote species. Anyways, what do we have here?
That's brain coral.
It's a brain coral.
Yeah. Lobophilia.
Lobophilia. Amazing. I love your work, guys. So this also has been recently in the last 5 years or so, changed. This one used to be called Symphilia, but now it's Lobophilia, which is an easier name to remember. So it's nicer for everyone involved. What's this one here?
Acropora.
Oh, so we had an Acropora, not quite right, but it branches and corymboses like Acropora does.
Astrophora.
Hydrophora. What is it?
I think two of you guys just said it then. Perfect. This one's Astrophora, which is transferred onto this part of this structure here, and they grow incredibly fast, unbelievably fast sometimes. Like, yeah, it shocks me still to this day.
A sawtooth one.
I'm acting them all out. So you guys should be looking at my screen because I'm...
Pachyceres.
Yes.
Tachy-
Ika.
Tachycyrus.
Yes.
I can't say
Zakia.
it.
Oh, everyone's nailing it. Love it. Pachyceres.
Polytest.
Porites.
Yes, 2 of you. I couldn't tell who that was them, but 2 of you guys just yelled out Porites, and that's bang on. Can anyone tell me what's happening in this photo?
Warfare.
2 species is
Warfare.
Yeah, warfare. We've got 2 of the same species. Oh, looks like 2 of the same species that are competing for space, and they're trying to grow over each other. And it looks like the top variety is winning. Awesome. Well, this is a tricky one because the tentacles are out, so we'll see how we go. Yes, Anthony. Pavona. Yeah.
Pavona.
Oh, I like that. I like that a lot. That was awesome. I hope you all say that in your head now every time you ever go to say Pavona. Awesome. What do we have?
Elouise.
Oh, yes, that was like 3 people. Yes. You said, if you said Goniopora, which is what I think you said, you'd be absolutely right. Awesome. So there's the 24 tentacles there. There's the other one that looks the same, or at least very similar, long tentacles, but there's 12. That's Elle Haskin. But that doesn't matter. You guys nailed this. I think this is the last photo, guys.
Acropora.
Yeah, I just heard it. A few of you once again. Yeah, awesome. This is Acropora. So good. Congratulations. You actually made Squidward smile, which apparently is quite rare. Yeah, I mean, I think we've all seen SpongeBob. So that was awesome, guys. Not too many of you have like logged out, and, you know, I hope you're not leaving this hating me and thinking, God, that was boring or anything like that. Have we got any questions or sort of anything like that? Are we feeling like we want to do the exam? Actually, maybe in terms of the exam, some of you have already emailed me. If you did already, sweet. If you guys seeing answers being written as well, all the right answers. If you guys haven't told me yet that you want to have the exam, just send me an email, because there actually was, you know, quite a few of you. It might just be easier for me to just have a list in that variety. If you do, I'll send it to you. It's predominantly multiple choice. It's a lot of like photos being like, you know, here's this photo. Is it this coral, this coral, this coral? You know, I'm not gonna, I don't mind if it's open or closed. You guys can decide if you use other resources or not. But, you know, it's testing yourself. So send it to me when you're done. I'll send it to you as a PDF. You can use, you know, Adobe or whatever you want to write on top of it, and I'll give you guys feedback if I feel like there's anywhere that you need more, whatever. I can let you know. Also, do let me know if you want like more resources. We've had some amazing students and interns and teachers in the past have just made some really, really cool resources, like little puzzles and little keyword things and just cool lists with like symbols and how to remember different coral. But yeah, that's like mostly it from me. If you guys haven't heard already, my boss Chad, CD founder, is going to be doing a series of talks on mineral accretion. Since you guys all did this one, if you want to do that, it's 20% off. No pressure, but he's awesome. So there we go. And literally, if any of you guys have any questions right now, like just say it. You can unmute, you can type. Oh, thank you guys so much. Yay, I'm glad you guys liked it. I hope you could tell that I was having a pretty good time. I love talking about corals. But yeah, really appreciate all of you guys being there. If you want to, you can start logging off. Oh, actually, could we get a screenshot? Could everyone smile? Because I just love this stuff. Yeah? You don't have to turn on your cameras if you don't want to. There's absolutely, oh, Jade, hello, oh, Alex. Ah. If you want to, though, that would just be so fun. Okay, 5, 4, oh gosh, some more are turning on. Oh, okay, I'll just wait for everybody in that case. Yay. Was I understandable, everyone? Was I a little bit too loopy, or was I sort of okay? Awesome.
Brilliant.
I hope you felt the excitement from my end. Okay, guys, 3, 2, 1, whee! Oh, thank you guys so much. I appreciate it. I'm going to quickly paste that here before I lose it. Yeah, feel free to log off. I loved having you. You guys were literally amazing.
I have a question. How are you guys doing under lockdown? You know, with everything that's happened in the world right now, how are you guys doing in terms of getting out to the reefs and doing your surveys, and also how are the coral reacting to people being out of the water?
Well, corals, so there was a heat event last year. It wasn't like a global event, but some of our sites were recording like some bleaching, which isn't ideal. Like I nearly, you know, like it's not nice, but the smallest part of me, and I think a lot of people thought, oh, well, if this is gonna take a little bit of pressure off of people being on the reefs all the time, maybe a little bit of good can come out of this. But of course, there are other factors at play here. In terms of our centres, how are we getting out into the reefs? So we have got instructors. So some of our sites are simply like the countries they're in, they are not accepting tourists. So in terms of like a program open for ecotourism and education and research, not much is happening, but we still have instructors at 3 of our centres, one that's more 4 if you include the one that's about to be open. They are pretty much catering to local like customer bases, so still going, but it is a much smaller level to what it's going at before. One of our locations isn't manned with one of our instructors just because of, you know, the circumstances. He stayed for as long as he could until he couldn't, and he's struck up a deal with some of the other conservationists on the island, essentially like taking them through the ropes, sort of said, you know, keep an eye on things as much as you can, and they were super wonderful about it. So things are definitely working at less of the capacity. We just want people to sort of remember, hey, we're still here. We're trying to have more of an online presence than we used to. Like obviously some people are saying, you know, like I wanted to do all this marine conservation, now I can't get out in the field. And like we feel that, like a lot of us aren't either. I'm not at any of our centres anymore. I was working full time in our centres for 2 years before essentially was coming home to Australia to say g'day before going back out, and then got stuck here. So I decided to go back and do postgraduate because I was like, oh, I might be stuck here for a while, so I may as well. A lot of people don't realise, but Australians are not allowed to leave Australia, so we're locked in. It's working well, but I physically can't go anywhere right now. Thanks for the nice messages, guys. But yeah, so we're pretty much just trying. We heard, like, we got questions from people saying, you know, I still want to do this course, or I still want to do this course. And yeah, unfortunately, if you're not at like centres, we can't do the diving components, but like we are so happy to provide the theoretical to at least still keep people, you know, in the mode of, okay, I'm learning, I'm still, you know, it's not just conservation. You don't just go out and pick up corals and that's conservation. You have to know the theoretical, you know, constraints and understanding before you go do all of that. I mean, this lecture, for example, to collect in-depth data, you need to have an understanding of coral taxonomy, you know? So even if maybe you guys aren't right now doing work, if you were to go into the field later, or if I were to go into the field later, this knowledge is essential for doing that whenever that does happen. So we're still just trying to remain present, even if it is online. We've also just launched a Patreon, which we kind of want to make a more cost-effective way for people to access like all of our stuff. We're going to be giving advice on everything from like some of the content like this, but also largely like advice, like how do people like you and me go out there and realistically start like a conservation or conservation diver program? You know, like what constraints might there be? Like what permissions might you need? What business models can be useful? Are you going to be a non-for-profit or a social enterprise, or, you know, whatever it's going to be? So we're trying to provide support in those sorts of ways, you know, to at least sort of give people something else to work on when they can't necessarily be in the field doing work. That was a long-winded explanation, but yeah. So we do have some stuff happening at our locations, but not as much and not all of them at the moment. It's just sort of what it is. Yeah. Anything else relating to corals, conservation diver, anything like that? I'm so thirsty after all that. Yeah. Cool.
I'm surprised you got any voice left.
Trevor, I can talk. I can talk forever. If you hadn't noticed already. Yeah. But yeah, really appreciate having you guys all here. I'm just going to hang around, like in case some of you guys want to ask some questions, like when there are less people logged on. So feel free to log off. Some of you guys probably have like lunch to eat or dinner to eat. I know there was a Jade is, I think, down the road from me, like in Perth, which is amazing. We've got to be really close. Perth is not a big place for everyone else out there. It's the most isolated, teeny tiny city in the world. But yeah, so anything else?
Well, I'm going to log off, Ellie, and I'll probably email you at some point.
Absolutely.
All
Awesome.
right, it's been great. Thank you so much for having me again.
No worries. Have a great
get enough
morning.
First time
Morning.
today.
Love
Bye.
you. Bye. Cool. Yeah.
Awesome. Right. Thanks for that.
No worries. Thank you so much for joining.
Yeah. That was awesome. We'll be in touch soon.
Yeah. Email whenever you need. Absolutely good.
Yeah. Lovely. Thank you.
Bye. Awesome.
Thank you. Bye-bye.
Thank you.
Bye.
Bye-bye. Ciao.
See ya.
Bye-bye. What's this?
Alex, do you want the exam? You're the one that initially inquired about having another taxonomy presentation. Only if you want to. I know you've done it before, so you don't have to. But if you want to test yourself again, obviously I'll send it through. You can do it as you enjoy it, as you want to.
Yes, please, if that's all right.
Yeah, of course. It's too easy. Awesome. Great.
Yeah.
Cool. Oh, just a couple of us now. Wonderful. Thanks for joining, Natalie, by the way.
Yeah, no worries. Thanks for the offer. It
Yeah,
was absolutely amazing.
Oh,
Yeah,
glad you enjoyed.
very
Cool.
much so.
All right. Okay.
Well, hope you have a good evening.
Yeah, you too.
Talk to you soon.
Bye. Bye. Okay. All right. Then Louise, I'm not sure if you're still there, but it's been really wonderful. Thank you so much for joining. I'm just going to read through some of these messages and then I think I'll probably end the chat. But feel free to speak or to text if you want, write on the thing. Yeah. Oh, some nice people. Oh, message. Hey. Hey, Louise. Thanks so much. Okay, so we don't send out the whole slideshows. We used to, and it's a little bit silly, but something like we did have a couple of people end up like sort of like, I know it sounds silly, but sort of selling it. But let me know, even if you want like a list of those corals, and I'm happy to send you sort of like my own pictures, but just sort of like with the whole setup with like our logos and stuff, I won't be sending you the whole thing. But yeah, if you want more photos, a list again, or even just like the family names, the genus names, so happy to send you whatever you want. If you want, just send me an email, I guess sort of exactly what you want in that regard. Like if those corals, I don't know if you're a diver or a snorkeler already, but if they were relevant to you and you thought, oh, actually this lecture was pretty reflective of what I've seen, yeah, just chuck me an email or send me another message right here and I'll send you through a list. Happy to send you some of my own personal photos. Nearly all the ones on these slides are compiled from our team, plus a couple of other which are credited, but like we've got so much that we can provide. So yeah. And also let me know if you want the exam as well. There's no like rush to do it. Like you don't have to do it right now. You can do it in your own time, but like so happy to provide some feedback on that too. So yeah. And thank you, so much for joining, Elouise. We've got pretty similar names. Mine's Elouise, so nice to meet you. Yeah. Oh, no problem. Yeah, I understand. Oh, not a problem. Oh, awesome, beautiful. Well, just if you do change your mind, you just like want some extra photos to work with, everything's Googleable, but just because there has been like a massive shift in, not a massive shift, but like, you know, taxonomy is changing and being updated. If you're ever like unsure, if you're seeing multiple things come up for one search, feel free to message like whenever you like, because I really do love this stuff if you can't tell. But yeah, I've had you for long enough. So yeah, I'll let you go, and yeah, feel free to email, and have a good morning, night, middle of the day, wherever you are. And yeah, have a good one. Awesome. Bye.